Yeah, thanks a lot for the invitation first. It's really fun to escape the freezing German weather and to come to this beautiful city. Okay, so first of all, just very briefly, I think all of you know that HIV-1 has been, unfortunately, an extremely successful virus that has meanwhile infected about 70 million people, and about half of them have died already. And we are studying restriction factors and the virus a bit from an evolutionary perspective. So just very briefly, the virus, to our current knowledge, most likely originated from a chimpanzee that transmitted its virus to a human about 100 years ago. And studies by Beatrice Hahn, Martin Peters, and others actually allowed us to obtain a relatively detailed picture on these cross-species transmissions. So meanwhile, there are 4 groups of HIV-1. One group is the so-called N group, and this group is only found in about 15 individuals, most of them living in Cameroon. And then actually the first sera infected with pandemic HIV-1 group M strains originated from Kinshasa, and the closest relatives in chimpanzees are found in this area in the Congo, which looks far apart, but actually often hunters basically hunt chimpanzees over here, and then they travel down the Congo River to sell the bushmeat in the city. And of course, it's only one of several plausible explanations, but handling of contaminated bushmeat with open wounds is at least, to me, the most plausible explanation for transmission. There are 2 other groups, O, that has infected about 100,000 people, and P viruses that are only found in 2 individuals so far. And surprisingly, both of these groups have their closest relatives in gorillas that were most likely also infected by chimpanzees. So HIV was very successful, and this is actually a bit surprising because all of us and also other mammalian species have evolved a number of antiviral factors. And the 3 best characterized ones are TRIM5 alpha, that basically targets the viral capsid to induce untimely uncoating. Then we have ApoBex3G, that is usually incorporated into viral particles and then deaminates the viral genome to cause lethal hypermutation and to inactivate the virus. And then, as another backup mechanism, we have tetherin, that acts as a late step and basically really acts as a physical tether to prevent the release of new viral particles. The list of restriction factors is increasing and could be made much longer. I won't talk about SAMHD1 because this is going to be addressed later in the session. One recently identified factor is SRANK5. That is incorporated into viral particles and drastically reduces viral infectivity. And one interesting aspect about these restriction factors is they're really highly diverse. There are no rules that apply to all of them, but there are some common properties that most of them share, which is basically that they are often inducible by interferons. They are frequently interacting with viral components, and they are usually under strong positive selection pressure for change to become active against new invading pathogens or to become resistant against viral antagonists. And we recently used these properties in collaboration with Amalio Telenti to perform genome-wide screens for cellular factors that share properties of known restriction factors. One factor that we identified was GBP5, which belongs to the interferon-inducible GTPases that have a really well-known role in innate immune defenses against various pathogens, also bacteria and others. So it's a protein of 67 kilodalton that has a GTPase domain and an isoprenylation site. And actually, a GTPase activity is not important for its antiviral function, but isoprenylation is. So what we found was that when we overexpress GBP5 together with proviral constructs, we lose incorporation of functional GP120 glycoprotein, and we get increased incorporation of non-functional GP160 precursor. And this showed that GBP5 interferes with viral infectivity and reduces incorporation of functional envelope glycoproteins. And of course, overexpression can always create artifacts, and that's often the case. So what got me excited was that when we just looked at the endogenous GBP5 expression levels in macrophages that were not stimulated just before infection with HIV, and the infectiousness of the variants that we got out, it actually correlated over about 3 orders of magnitude. And this was getting me really, I thought, wow. But of course, this could just be coincidence, because it could just be an indicator of general activation of the macrophages, and it doesn't prove causality. Although when we did knockout studies, we found that actually about 60% to 80% knockdown of GBP5 increased the infectiousness of the variants that we got about 5-fold. So it's not only correlation, but it really seems to play a major role in infectious virus production by macrophages. So this just summarizes this. We found we had much more data on it, but basically they showed that GBP5 interferes with various steps of envelope processing and incorporation into variants, and really drastically reduces viral infectivity. Now, one question is, of course, if there are so many antiviral factors, why do we actually have a positive correlation between interferon levels in patients and viral loads? So why do they not really work and control the virus? And of course, one thing is that HIV can rapidly become resistant, and that's, for example, the case against TRIM5 alpha. So the HIV-1 capsid is very susceptible to TRIM5 alpha from monkey species, but not from humans. In other cases, it's more difficult for the virus to become resistant because it cannot live without or replicate without its DNA or without its envelope. And therefore the virus has evolved specific antagonists, like the virus infectivity factor, that basically targets ApoBec3G for proteasomal degradation and destroys it in the cell. And for pandemic HIV-1 strains, that's a bit similar with tetherin, that is targeted by the viral protein U. And I have to apologize that I forgot to put this in, but basically tetherin on its way to the surface is also degraded in the proteasome. Finally, the most recently discovered restriction factor serine, this was actually cheating a little bit on you, because this inhibition is only observed for viruses that lack the NEF protein, because the NEF protein very efficiently counteracts serine-5, and it does so by removing it from the cell surface, so that's the normal localization, and then this is what you get in the presence of NEF. So one take-home message here is that HIV-1 has evolved like a variety of tools to get rid of these antiviral factors, but you should really memorize them as more like this. I don't have time to talk all about all their activities, but they act more like Swiss Army knives, because they are multifunctional. So if they are inactive against HIV-1, are they good for anything at the moment? And I think many of you know this red-queen hypothesis. So basically the arms race between between pathogens and their hosts, which basically means if you have an antiviral protein, first of course it will work against the viral target, but then the virus becomes resistant, the host adapts, virus becomes resistant again, and this goes on, and this is one of the reasons why for this high positive selection pressure. An important thing is that basically this antiviral protein will not work anymore against the virus that emerges at a later time point. And therefore, many of these factors act in a species-specific manner. And this species specificity is most likely one important reason why only 3 chimpanzees, gorillas that I should show here, and Sutimanga-bees have transmitted their viruses to humans, and the other about 40 species in Africa that are infected with these viruses have not transmitted their virus. So actually, when we look at the prehistory of AIDS, it turns out that there were actually quite a few cross-species transmissions and also adaptive events that preceded the AIDS pandemic. It's by now well established that chimpanzees received their virus from red-capped mangabees and the precursors of these Cercopithecus monkeys, on which chimpanzees prey, and this is most likely how they became how they became co-infected. And then later, this virus was transmitted from the chimpanzees to gorillas and to humans. So actually, the big jump for these viruses was not from the great apes to humans, but from the small monkeys to the great apes, because they are genetically more distant related. And the barrier here was basically all the different restriction factors, ApoBec, TRIM, Tetherin, because of the species specificity. And what actually happened was that these 2 monkey viruses recombined to generate a new virus that became the precursor of the AIDS virus. And during this recombination step, the virus lost its VPX gene, but it generated a functional VIF gene, and also a few other things happened. But basically, there was a new combination that worked at least to some extent in chimpanzees, and was a prerequisite for the later evolution of the AIDS virus. So adaptation to chimpanzees then already inactivated a lot of our defenses, because the chimpanzee virus is already fully active against human ApoBec and against human TRIM5. Now, one question is then, of course, why have these viruses still spread to such a different extent? And of course, there are many factors that play a role. It's very simplified because when I focus on restriction factor. But one important factor that may have played a role here is Tetherin. Now, I really like Tetherin because most things in biology are so complicated, right? With Tetherin, it's really nice. One end sticks in the virus, one end sticks in the cell, and then you have a helice. So it's really like, basically anchoring the virus, nice and easy. And because it's such an easy principle, this restriction factor also works against essentially all enveloped viruses that bud from the cell surface, so very broad activity. And as I already said, pandemic HIV-1M strains use a VPU protein to counteract Tetherin, and VPU interacts with Tetherin, that is also called BST2, in the transmembrane domain by direct interaction. And I already said that these factors are multifunctional, so VPU also degrades CD4 and counteracts Tetherin. And in the absence of VPU, HIV-1 just sticks at the cell surface and can only get away if this factor is counteracted. So what we found at some point is that only the VPU protein of group M viruses really has evolved an active tetherin antagonist of human tetherin while maintaining the CD4 down modulation function. N viruses have only acquired very modest anti-tetherin activity and do not degrade CD4. And O and P viruses do not use VPU, cannot use their VPU proteins to counteract tetherin. So that suggests that really only the pandemic viruses are optimally adapted to humans, and this could, of course, play a role in their efficiency to spread. Now, why was it so difficult for the virus to become active against human tetherin? Most SIVs, including SIV from chimpanzees and gorillas, use their NEF protein to counteract these restriction factors and interact with the cytoplasmic domain. Now, humans have a deletion exactly in this area that is targeted by NEF, and this renders the human tetherin autolog resistant. And actually, this deletion is already quite ancient and already found in Neanderthals and Denisovans, so it already emerged about 1 million years ago. And this is now the oldest. I'm gonna go back. We studied tetherin autologs from ancient shark and other ancient fish, and they already have active tetherin. So the principle is more than 500 million years old, which shows you like how long this battle is going on. So, as I said, these chimpanzee viruses switched to VPU, and group HIV2 actually switched to from NEF to ENV to counteract tetherin. And what we also find is that, for example, group N viruses are still adapting to humans, because actually the most recently transmitted group N virus that was transmitted in Togo and and discovered in France actually now antagonizes tetherin as effectively as VPU proteins of the pandemic strains. And we also found, and sorry, this looks a bit complicated, that group O viruses were most likely capable of infecting about 100,000 people, because they managed to evolve NEF proteins that now interact with a different region in tetherin, more to the N-terminus. So this region here that is missing is targeted by the gorilla virus NEF, and then in the humans it switched to this area, but it cannot counteract tetherin as efficiently as VPUs. It does it a bit less well. Okay, so in summary, why is the virus so successful? It basically can either rapidly become resistant against antiviral factors, or it has evolved specific tools, the accessory genes, to counteract them. And it's actually amazing how flexible these accessory proteins are. And this is perhaps also illustrated by the fact that the virus cannot only use them, but even switched between NEF and VPU-mediated tetherin antagonism several times during its evolution. Don't have time to go into the detail, but so they can also jump in basically for one another. So final, and maybe it's noteworthy that without the group M viruses, AIDS would still be a rare tropical disease. And the last slide now, perhaps these restriction factors even play a role in the spread of SIV in the wild, because what we found recently was when we look for the efficiency of NEF to counteract serine-5, and we look at the seroprevalence in great apes or in monkeys that is very variable, we actually see a significant correlation. And it may make sense that the release of the virus into genital fluids due to tetherin antagonism in combination with its infectiousness could of course play a role. It's plausible, but it's also just indirect evidence, and basically definitive proof would need further investigation. So with this, I'd like to thank my many collaborators, particularly Beatrice Hahn has an essential role on this, and we are collaborating for many years and exchanging reagents and ideas. In my lab, Daniel Sauter and Dominic Hotter had an essential part in the work. And we also have very nice collaborations with Martin Peters and others. Thanks a lot for your attention. Happy to take questions.
Are there any questions? Please.
Thanks. Excellent talk. What are your thoughts, not so much between populations, but within individuals? Because Beatrice, as you note, has been describing this interferon resistance in acute infection relative to what would be the initial pressure of interferon-mediated gene expression, at least in the context of acute infection. But then she describes that these isolates sort of then are not as prevalent in chronic infection. So do you think that this selection for being able to break through all the interferon-mediated resistance factors is optimally pressured by the host at the beginning and then sort of retreats back, and that's why you would not have resistance against all of them unless you're under very extreme pressure? I mean, in the context of the within host, how much variance do you see between the pressure to keep all the resistance factors active?
I think there's always some pressure, but I think the virus is basically, because it can adapt so rapidly, is a master of compromise. So it basically just does what it has to do. And when you look at the transmitted founder viruses and the chronics, it's not that they are really separated entirely. It's just they have a tendency, the transmitted founders, to be more resistant against interferon, which we can also see basically in our assays. And I think definitely, I mean, the interferon induction, innate immune induction, is highest during the acute phase of infection, so there it will be more important. You only have adaptive immunity at later stages, and there are now also, I mean, for example, you most likely saw this nice recent paper from Stuart Neal's group showing against phytams that actually initially the transmitted founders are resistant against them, and then they become vulnerable because they accumulate escape mutants against antibody and CTA responses that then make them more vulnerable. And I think this may well also happen with other restriction factors. Which was most striking to me is that Beatrice sees consistently this differences in susceptibility, but they map in regions of the virus frequently that suggest the presence of further restriction factors that we don't know. That's for me also one very interesting aspect.
Excellent talk. I am a clinician, and my question is, do you think that the interaction between the BPU Cetherin could be a new antiviral target, or just is in order to understand the function of these restriction factors?
I guess the thing is that we have excellent drugs at the moment that work very well, and these effects are more modulatory. So even if you have lack of BPU entirely, the virus can still replicate to some extent. And also I think even if you block it, it would help to some extent, but it would not be as effective. Although, I mean, people, for example, have made transgenic cats for trim, and they are entirely resistant. So with other restriction factors, this could certainly be.
No, I was thinking in the example of the influenza virus and the junction between the neuraminidase, the hemagglutinin and sialic acid, and the role of the neuraminidase. No, stopping the inhibition of the neuraminidase, you can avoid to release the virus into infect another cell. So I don't know if the Cetherin has the same.
No, I think it's quite different. Because, I mean, Cetherin is pretty unspecific, actually. It targets not so specifically the envelope, but just the membrane of the virus.
Thank you, Frank, for this talk. I have a naive question. The deletion in Cetherin that we observe in human in comparison with chimpanzees, do you think that the retrovirus infection could be a pressure for the selection of this specific deletion?
Yeah, it could well be. I mean, some of you may know that actually they found endogenous lentiviruses in rabbits and in lemurs that already entered the germline like several million years, 6 and 8 million years ago. So these viruses are around for much longer than we thought. And this lentivirus already has a VIF gene. Although Cetherin also works against herpes viruses. So whether or not it was an ancient retrovirus or another virus is impossible to say.
Thank you.
Two additional questions by the chair. One very specific, GBP5. Is this specific effect on macrophages, or you see it all throughout CD4 T cells and other cell types?
It's difficult to tell. I mean, basically, I worked more on macrophages than I ever wanted to because we cannot do knockdowns or efficient knockdowns in primary T cells. So the factor is also expressed in T cells. We are working on it to see if it also plays a role there, and this is ongoing work. Now when we express it in primary T cells, we get inhibition, but we don't have clear-cut data what happens when we knock down the endogenous factor.
The other question is a broader one. I'm just trying to be a bit counterintuitive on restriction factors. I mean, the fact that, you know, there is sort of like a tug of war between the immune system and viruses. Would it be possible that restriction factors actually are not beneficial to the host?
Why not?
Well, because you may see an overriding viral replication in which containment doesn't come from particular cell factors, but by the immune system as a whole, in which you don't need to actually limit replication. So you could mention a few restriction factors that in fact may be elements that induce inflammation when you don't want it.
Yeah, for me, it's actually that the restriction factors are more a consequence of inflammation, and I mean, they also can act as sensors to some extent, so you're partly right. But personally, I think that they still do a good job, because I think, like, in most cases, they will protect us. I think these transmissions that we see nowadays are just the tip of an iceberg, and I mean, humans were exposed also to these lentiviruses for many thousand years, and I think that only a very few of these exposures were successful, and there were like a number of unfortunate things that made the pandemic M viruses so successful. So I think most likely they work in 99% of all cases, and what we see is only like what didn't work. So I think they are good for us, and maybe it's also suggesting that I think to some extent they still help us to control the virus. I mean, there's evidence for that. They're not perfect. That's the problem. And I think in the case of HIV, it's unfortunate, because there you may in part be right, because they may contribute to the inflammation that is harmful. But I think that's really a very unfortunate exception.
All right. Well, thank you very much, Frank.
Thank you.